I'm going to walk you through some of the steps of gametogenesis today. So there are some other videos you can watch that'll walk you through this, and fertilization. There are a million videos on the internet. If you need anything to supplement your understanding, and some of this may be old information from another biology class. So decide what your needs are and how much information you need to understand this topic. So fertilization is the union of sperm and egg. And you have to remember that, again, the sperm cell and the egg cell only have 50% of the information to make a person, which is why we need to put them together to make 100% of the information needed. Now, the process by which we make egg and sperm, that's gametogenesis, and that's what we want to walk through today. And they are similar processes, but there are some really key differences between the two. Again, if you need to go back and review what goes on in normal somatic cell division, which is called mitosis, please go back and watch those videos or read through that portion of the textbook. In normal cell division, we are producing two brand new cells that are identical, right, to the original parent cell. Meiosis is what we're going to talk about today, because that's how we make the gametes and do gametogenesis. So this is the process by which we don't just make two identical daughter cells, we actually make 4 cells, and they're not identical because we split the chromosomes up differently. And those 4 have half of the amount of information that we need. There are 2 stages so that we can divide 1 cell into 2 and then 2 cells into 4. So we actually go through a meiosis 1 and then a meiosis 2. Meiosis 1 is very similar to mitosis, except for the fact that when we pair up our chromosomes, they get sorted randomly. So we get a mix of mom and dad information into those 2 cell products. This is where we're starting to create some variety. In meiosis 2, we don't have any sort of DNA replication, so this is where we're really doing what's called the reduction division, and we're cutting the amount of DNA in half. And then there again are a little bit of similarities back to mitosis, where we're going to break apart sister chromatids, but again, we've already done some of the mixing up and variety generation in meiosis 1, where we shook up and mom and dad's chromosomes and we didn't know which ones were going where. What's critical is we maintain a constant chromosome number from generation to generation. It's advantageous because, again, we're getting genetic variety. So if you have someone in your family that is sick and has a disease, there is not a 100% guarantee that that disease will always be passed on. So maybe some of your offspring will get the disease and some won't. And again, we have variety there so that we end up being a healthier population. If you weren't able to do meiosis, say you were a banana and you had like 3 sets of chromosomes and you aren't able to divide them evenly, we would not have that. Bananas really don't have much of a genetic variety. They are delicious though. Okay, so if we line them up, here is mitosis in the brown panel, and here is meiosis in the purple panel. So let's compare and contrast these. Again, you're not ready for this if you haven't watched some of the other videos that are available, or if you haven't had a general biology or a genetics course recently. So you might want to pause this video because I'm just going to get right to it if these terms are not yet familiar to you. So again, the big difference here between mitosis and meiosis whenever we go through this preparation for cell division, right, in this first panel here, is in mitosis, right, what's going to happen is we really need to pair up and match up all of our chromosomes in pairs, right? We're replicating them, and then what we're trying to do is evenly divide, right? So if you follow the blue chromosomes and the orange chromosomes, we see at the end of this, okay, we end up with the same number of blue and orange chromosomes in each cell. If we go over here in meiosis, we have our homologous pairs lining up. So let's say mom gave the blue chromosomes and dad gave the orange chromosomes. These guys are going to pair up and find each other, and they're going to get pulled into different cells. And again, there's no guarantee here that all of the blue ones from mom will go into one cell and all of the orange ones from dad will go into another cell. Also, we get this little bonus thing of crossing over. So the chromosomes are so close, they're touching, and sometimes they kind of shake hands and literally break off little bits of them. So it'd be like if I popped my hand off and gave it to my neighbor, and they popped their hand off and gave it to me. And so the chromosomes can swap little bits. That increases the genetic variety even more, because now it's not just one whole chromosome here and one there, right? Now we have little swapping. Okay? And that's if you have siblings and you see that some of you take more after mom or dad and some of you really are quite the mix, crossing over, right? You might have had different amounts of that. Okay, so again, whenever the chromosomes split up, we can see the little crossed over bits. And this one did kind of work out where it's mostly a blue chromosome cell and mostly an orange chromosome cell. And then we divide one more time to go through meiosis too. And again, we end up with half the amount of chromosomes and twice the amount of cells. Okay, so again, there are really two big ways that we get variety. I've already indicated that, but if we need it to be spelled out just one more time, let's clearly state that. Again, we have this idea of random assortment. And now the chromosomes are not blue and orange, they are blue and purple. So again, they are color-coded to indicate one is coming from mom and one is coming from dad. And we don't know if someone is going to independently assort them into a mom cell and a dad cell, or if it's going to be random, right? We don't know which thing is going to happen whenever we're making a particular sperm or egg cell. Again, this is why you and your siblings or other relatives share some features, but you won't share all of your features. And the crossing over I mentioned, this is the idea that the chromosomes get so close to each other that they will end up swapping little bits. So now we've got a little bit of the blue chromosome on the purple and a little purple on the blue, again, creating a little bit more variety and a little bit of mix of traits from mom and dad. Variety is good. This makes us stronger as a species. If some disease came and wiped out all of the people that have the purple chromosome trait, we would be happy that there are some people in our family that have the blue chromosomes, and maybe that some of the people with purple chromosomes maybe got a little crossover bit of that helpful thing from the blue. So this is good for variety and survival of the species as a whole. Not good for the individual that got the bad purple chromosomes. All right, so let's do this once for spermatogenesis and then once for oogenesis. So we have a couple of cell types that we need to introduce. With spermatogenesis, we are going to start with spermatogonia. Remember, whenever we're in the seminiferous tubule, maturation of sperm cells goes from out to in. So we're going to start in this outer layer. This is where all of the spermatogonia will live. And these guys are the mitotic cells. These guys that are really are dividing and just producing two clones of themselves over and over again. But we know the cell division and sperm maturation produces from out to in, so we're going to move this way towards where we see our products, our sperm cells getting released into the center of that seminiferous tubule. And as they're moving through, they're going to start the mitotic division. So some of those spermatogonia products will become what we call primary spermatocytes. Okay, those are the ones that are going to start the first meiotic division, right? This is where we have the independent assortment and the crossing over happening. Okay, and then we'll end up with the secondary spermatocyte, and that is going to go through the meiosis too. Okay, as we go through the meiosis too, again, we've got these random chromosomes. They didn't color code them in this figure, they just made them all black. Okay, but then we end up with 4 identical spermatids. Okay, and then the spermatids will go through that process of spermiogenesis, where they eject most of their cytoplasm, they make their tiny little head, and they grow their nice long whip-like tail, and then they're ready to roll through the male reproductive system. You can go back and watch that first lecture if you need a refresher on that. Again, this is the spermatozoa anatomy. This is the final product. Basically DNA with a propeller. That's the insensitive nickname that we give the sperm cell. Okay, and again, I've already covered spermiogenesis in the first lecture in the male reproductive system, but again, this is just the process of turning that spermatid into what we recognize is that very dynamic design of the sperm cell that has a tiny bit of cytoplasm and that long tail. Okay, and again, this process is going from out to in. Right? We can see, right, our sperm cells and their tails getting ready to be released into the seminiferous tubule. Okay, now let's do oogenesis, and I will tell you where the rules are different. 1, we get a really uneven division of cytoplasm. So instead of ending up with 4 perfectly healthy gamete cells as we do in spermatogenesis, we only get 1 egg. We do end up stashing the extra chromosomes in what is known as polar bodies, but because the sperm and egg cells have to work together to create the zygote or embryo, they are a little bit different. The sperm cells have a long distance to travel, so it's important to make a lot of them because some of them are going to die along the way. And again, it was important to make the amount of cytoplasm that they're carrying minimal because they're going to have to swim. They want to have a light load to carry. They don't want to get tired and die on the way. Now, the egg doesn't have very far to go. And the egg needs to, instead of getting rid of all of its cytoplasm, it needs to compensate for the lack thereof in the sperm cell. So it's going to be really big, and it's going to take an unfair share of the cytoplasm. So that's why we end up throwing away these other little cell division products that we're making in meiosis 1 and meiosis 2, the first and second polar body. These may have some other function in other animals and plants, but for humans, these things just get kind of thrown out in the wash. The other thing that we're going to have to take into consideration is when is this happening. So for sperm cells, spermatogenesis is a 24/7 process that is happening in reproductive years. So pretty much from puberty until death in men. Maybe you're making a few less in your 70s and 80s, but you're still making some number of sperm cells. In women, right, we kind of have a clock that we're turning on and turning off a couple of different times. So again, let's just walk through this before we get to the timing of it. And again, point out that, you know, there are the same type of cells. So in answer to spermatogonium, we have oogonium, right? Those are the mitotic cells. And then eventually some of those daughter cells will turn into primary oocytes, matching the primary spermatocyte. But again, whenever we start going through that first meiotic division, we end up with 2 secondary spermatocytes, but 1 secondary oocyte and then 1 polar body that gets the unfair deal of not getting enough cytoplasm to be worth our while to keep around. So then we go through meiosis 2. And again, we get our nice 4 spermatid products. And again, we're going to keep 1 good ootid, and we're going to toss our second polar body. We only have to make the polar bodies because we got to get rid of those extra chromosomes. It does us no good to keep those around. Okay, so again, the answer to oogenesis is a single egg product. Okay, so now let's work in the timeline. Again, men, this is happening 24/7, puberty throughout reproductive years. Ladies, oogenesis is happening differently. All of the mitotic divisions are done before birth. So, a female human baby is born with all of the eggs she is ever going to have. That's your supply. That's, you get what you get. Our oogonium are not active like they are in men. And once you're born, that's it. They're done. So we have all of the primary oocytes that we're going to need. Okay, these things are ready to go. Now, those primary oocytes, while we're still in utero, while we're still doing our fetal development, we're not even born yet, they are going to start meiosis 1. So they will start to divide and get that process underway. And then they stop. And they're going to wait for puberty. So they're just going to be like, all right, we're ready for meiosis 1. And hang on, let's wait 12 to 15 years. Or longer, because you're only going to do 1 at a time, right? Some of those have to sit there and wait for 40 years or 50 years. So they're going to stop. At menarche, okay, 1 batch of follicles at a time. So the shortest amount of time one of those primary oocytes is going to be waiting in meiosis 1 is, again, puberty, like 11, 12 years. The longest one of them is going to wait is, again, like 50 years whenever we get to menopause. So some of them are waiting different amounts of time. Then it's going to stop again. Okay, so we get through, right, and we're rolling through our menstrual cycle. So we're getting our meiosis 1 done, and we're starting meiosis 2, and then we're stopping. And what we're going to do is we're going to stop meiosis 2, and we're going to wait for a sperm cell. So ladies, most of our secondary oocytes never even finish the process of making that single finished ootid product. If there's no sperm cell, they're just like, meh, is it even worth it? Right? And it's not. So if they're not going to get fertilized, they don't even really care to finish their meiosis. They're just going to die and leave the reproductive tract, and there's no point to it. So the only ones that are going to finish their meiosis 2 and really finish making that polar body and that proper ootid, which is going to become the ovum, that is only going to happen in the case of fertilization. If you need to pause and read through any of the text on this figure or go back to your textbook, go right ahead and take your time with that. Okay. So again, just to kind of recap, The next lecture that I believe you'll watch is one that kind of talks about chromosome aberrations, and whenever chromosomes don't get divided evenly, that can lead to some problems with birth defects, or even not even be able to create a viable pregnancy. So it's really critical that these chromosome divisions happen properly. And this starting and stopping of oogenesis early in development in females, and then starting in at puberty, and then again, you know, some of those eggs have been sitting around for 50 years, that's often pointed to as a potential source or reason for not being able to produce a viable embryo. There are other causes too, that is not the sole cause, but this is definitely something that you want to think about. Okay, that is enough for this lecture.